All right, so today we're going to continue our lecture series, and we're going to be talking about another cell type. We've mentioned it before, and we're going to go into more depth today, and that is the topic of prokaryotes or prokaryotic cells. And so earlier we discussed that you have two main cell types. We have eukaryotic cells, those that have a nucleus and internal organelles. And then you have prokaryotic cells. Now, prokaryotic cells can actually be divided into two different groups. We have the bacteria. And originally we thought that all prokaryotes were actually bacteria, but we found out that they divide into two different what we call domains. And the second domain is archaea. And so prokaryotes are then, we can define these as single-celled, and the fact that they lack a nucleus or internal organelles. And so both of these groups from the outside have both of these characteristics. They are single-celled and they lack a nucleus. And for a long time we really thought that they were both the same group. Or that archaea was a subset of bacteria. And it was only when we started looking internally at the biochemical level that we saw not only some differences, but some really major differences. And so one of the first experiments that we set up, or that we started to identify the difference between these, was having to do with ribosomal RNA sequence comparisons. And so we learned earlier about the ribosomes, that the ribosomes are made out of a combination of protein subunits and RNA molecules. And so one of the things that they started to do, there was a scientist by the name of Woese, and he started looking at 16S. There's several different pieces of ribosomal RNA, and so they're designated by a number with an S in front of it, S being the units of that, Svedberg units. And so he was in particular looking at the 16S ribosomal RNA. And as he looked at the ribosomal RNA from bacteria and started comparing it with that of archaea, he started noticing some real differences in the sequences of those, even though they were performing basically the same function. And actually the comparison of ribosomal RNA is one of the methods that scientists use to distinguish between different species. Almost all organisms have ribosomal RNA, and because it's hypothesized that we descend from a common ancestor, what we find is that the more related two species are, or two organisms are, two families, the more they have in common on those sequences. And the farther apart those areas are, or those two species are, the more time has passed for mutations to crop up in those RNA sequences as they have diverged from each other. And so the farther apart two things are, the farther back they had their common ancestor, you'll see more sequence variations. And the closer they are, you see fewer sequence variations. And that was the idea behind the comparison of these ribosomal RNA sequences. Everyone had them, or all organisms have them, and when they started to compare them, you can start to tell how far those two species diverged. So as he was looking at these 16S ribosomal RNAs, he was really expecting to see a lot of commonality between the archaea and the bacteria, that they knew they were different organisms slightly, but they thought that they were really quite similar and that there'd be a lot of unity between those RNA sequences. And so he was probably, if to put it in terms of modern day human languages, he was expecting to open up the ribosomal RNAs as if each one were a book. If you took the bacterial genome and it was a book, and the bacterial genome and it was a book, and you looked at the sequences, he thought it'd be opening, a page of maybe Queen's English, British and a page of Yankee English, American English, right? And so you look at it, you see some differences maybe in the wording or how they explain things, but overall you could tell that they were written in essentially the same language. And what he found when he opened those two books and looked at the page, the ribosomal RNA page, he found that it was actually more like looking at English and Chinese, that there was absolutely no relationship between the two. And so he then started looking at more bacteria, and as he looked at different types of bacteria, he got what he was expecting, that English to English, maybe a little bit, few different words, but still the same language. And then when he looked at the archaea, it was like each one of them was written in that Chinese. And so you could group them based on the language, but there was no unity between the two. And so that led to the idea of different domains. And so he said that there's actually bacteria, archaea, and then we've already talked about eukarya, the eukaryotic cells. And we knew that eukarya was very different from bacteria, but he said archaea is as different from bacteria as eukarya is from archaea or bacteria. He said these are three different domains of life, and it really changed the way that we started to look at these single-celled organisms. As we started to look at them, again from the outside, these look very similar to each other. It was as if they were the same organism. So they're small, they're single-celled. Both archaea and bacteria have the presence of a cell wall. We'll talk about the cell wall more a little bit later in the lesson. Eukarya, you have animals that don't have a cell wall. But you have plants that do have a cell wall. So it seemed like there was at least a representative in each domain that had a cell wall, but when you started to look more closely at it, they started to diverge in that the cell wall of bacteria was always made from a chemical called peptidoglycan, a mixture of small, short amino acid chains and polysaccharides. And then archaea also had cell walls, but they were polysaccharide in nature with some protein subunits. And then you have the plants of cell walls, which are also polysaccharides. And so it was only when you started looking very closely at the biochemical nature of key components of the cells that you started to see differences. And what they did was they started then to look at these biochemical differences. And they found that in many ways, even though archaea was small and single-celled and lacked a nucleus, so it seemed like it was more related to bacteria, when they started to look at the biochemical processes, things like replication, translation, transcription, they found that it actually looked a lot more like some of the eukaryotic processes. And so archaea actually became not really a missing link, but seemed to have characteristics that are intermediate. And that somewhere along the development of the eukaryotic cell line, you had an ancestor that was shared with archaea. And so what are some of those biochemical differences? A lot of them, there is a feature in the genome of bacteria. There's something called an intron. And I'll briefly explain over here what an intron is. So if you have a stretch of DNA, so we'll label this as DNA, and between this cross and this cross, we're going to say that this stretch of DNA is what can be used to make a protein. So that has the instructions, and so we call that stretch a gene. So one gene is the region of the DNA that's used to make up a protein. And in prokaryotes, in bacteria, what we see is from the start of the gene to the end of the gene, it's contiguous, and everything inside there is actually used in the production of that protein. It's all useful information. So this would be a prokaryote gene, or sorry, a bacteria gene. Now, when you start to look at eukaryotic, we'll draw the same stretch of DNA, and it also makes a protein. The genes are often larger. But what they started to find was that interspaced in the middle, if the black was what would make the protein, they would find these loops of additional DNA. And these in blue were called introns, or intervening sequences of DNA. They wouldn't be used in the protein. So when the mRNA was made out of this gene, these blue sequences would be cut out and you just have the black sequences. And so you'd end up with the same protein either way. But you would have these intervening introns throughout the middle of the gene. We're still trying to figure out. We believe that there can be some function or purpose to these introns with alternative. There's things called alternative splicing that you'll learn in genetics. So there's a reason why we have these, but this is just a more complicated, advanced form to carry your genetic information in. Bacteria don't have it, and eukaryotic cells do have it. We knew this. We've known this for a long time. But it was only when we started to look at the archaea that we found some very basic introns. It's not quite the complex structure that we have in our eukaryotic genome, but they do have it. And then we have introns in the eukarya, of course. Another biochemical difference that we started to see when we started looking closely is in all eukaryotic cells, the beginning amino acid for all proteins in a eukaryotic cell starts with the amino acid methionine. So we'll call that a met starter. And this is true of all. See, in some proteins that methionine starter gets cut off, but they always start with the same amino acid called methionine. Well, bacteria also all start with a single amino acid. And it's similar, but it's also different. It's called F-methionine or formal methionine. So F-met is the starter in bacteria. And it's fundamental. So if you give a eukaryotic F-met, it won't be able to make proteins. And if you only give a bacteria methionine, it won't be able to make proteins either. It's fundamental that they have these two starter amino acids. Well, in archaea, if it's more like bacteria, if it's resembling bacteria, it'll start with F-met. The proteins will start with F-met, right? And if it's in eukarya, it'll start with methionine. Well, they have a methionine starter. So again, we look at topologically on the outside, from a distance, it looks like bacteria or archaea more closely resemble bacteria. However, when you start looking at the biochemical features, then they start to more closely resemble eukarya. We also see some differences. One way in which they're unique, so these guys have a plasma membrane. And the phospholipids that make up the plasma membrane are quite unique. And right here in the archaea, these guys have actually special phospholipids. So they have different membrane lipids that make up their membrane. Okay, and so that's another biochemical difference. So between these three domains, we find that archaea is somewhat intermediate. To wrap it up, they have significant biochemical differences, which was first observed comparing 16S RNA sequences, and then later looking at some of the biochemistry, it was confirmed that these are very different organisms. Again, as a different kind of analogy if you've ever watched Star Trek before. And so you might say take a human from Earth, or a Terran, I guess they're called, and then if you took a Vulcan, on the outside, other than the kind of the ears, it might look like they were very similar, and then, or even a Klingon, and then you open up and you find that the physiology is all different inside. That really, they're not closely related at all. Okay? And so that's what we're looking at those differences between them. Now, let's look a little bit more closely at the bacterial cell and some of its structures. So we did this in a previous lecture with our eukaryotic cell and what went on inside. And so for the bacterial cell, what's nice is it's a smaller cell and it's not as complicated. So we'll start like we did last time, drawing a cell. And then there's a few structures that we're going to draw. We'll explain what each of these structures are. So, as we draw the bacterial cell, the very first thing that we notice, like we did with the prokaryote or with the eukaryotic cell, we have a plasma membrane. So again, that's the outer membrane that separates inside the cell from the rest of the environment or the world outside of the cell. And so bacteria do have a plasma membrane. And then inside of that plasma membrane, they are going to have their chromosome. Okay? And the chromosome, one of the things you'll notice, so this is their DNA. And the chromosome does not have a membrane surrounding it. Remember, there's no nucleus in it, like there was in our eukaryotic cells. So we can't say that this is a nucleus at all, but the DNA inside the cell does tend to cluster in kind of one region. It's packaged in there, and it's all twisted up and in there. And so this is often referred to as the nucleoid region. Okay, so prokaryotes do have a nucleoid. They don't have a nucleus, they have a nucleoid. While we're at it, let's talk about a few things about prokaryote DNA. Just as kind of we did before. So, prokaryote DNA is different from eukaryote DNA or chromosomes. So in prokaryotes, you'll usually have a single circular chromosome. And you may not have noticed this when I was drawing it, but I started up here and then I drew and scribbled, and then I made sure that the two ends of my scribble connected. And that's because the bacterial chromosome is circular, makes a big ring. If you were to stretch it out, you'd see it was a big loop. And the prokaryotes will usually just have a single chromosome in them. In comparison, eukaryotic DNA usually has multiple linear chromosome pairs. So in eukaryotic organisms, the chromosomes tend to come in pairs. So you'll usually see even numbers of them. For example, humans have 46 chromosomes, which is because they have 23 pairs. Right? So in eukaryotic organisms, you'll have 2 copies or a pair of chromosome number 1. You'll have 2 copies or a pair of chromosome number 2. And those chromosomes are linear, so they look like long pieces. Sometimes we see them in this X structure that looks like this, or you might see it something like that for the human chromosomes, where this has been duplicated. But the point is, this is one single long piece. It's not circular, it's just long and linear. The size is quite a bit different too. In prokaryotes, you'll get a genome. Now we're talking about size. So a typical genome size might be 1 million to 10 million base pairs. So 10 million base pairs would be a pretty good sized genome for a bacteria or for an archaea. On the other hand, eukaryotic genomes, using our humans again as an example, we estimate is somewhere around 3.2 billion base pairs. Okay, so quite a large order of magnitude. Even simpler organisms like yeast have a total, I think they have 4 chromosomes, and it would be somewhere in the 100 millions. Okay, so the eukaryotic genomes, the sum total of all the DNA inside the cell, is much larger than the prokaryotic genome that's contained in the nucleoid. Other than that, we don't have, so we have the nucleus in here. Right here, all through this region is like it was in the eukaryotic. This would be called the cytoplasm of the prokaryotic cell. We also refer to it as the cytosol, being that fluid that's in there. The only other really classic organism or organelle that the bacteria have is one we learned about. It's not membrane bound. But of course, the bacteria have to make proteins, and so they have ribosomes in them. And there'll be a lot of different ribosomes inside the cytoplasm. We don't have any bound ribosomes. We don't have a rough ER like we do in the eukaryote because there's no membrane internal to the cell. But we do have ribosomes. They do differ from eukaryotes. The ribosomes in the prokaryote are actually different than eukaryotic ribosomes. They're smaller, for one thing, and they're just set up, they operate pretty much the same way, but they're set up a little bit differently. In fact, the bacterial ribosome is in many cases a target for antibiotics, where antibiotics are chemicals that kill bacteria, so they're anti-bacteria. And we use drugs that will bind to and inhibit the prokaryotic ribosome, but don't bind and inhibit the eukaryotic ribosome because they're different from each other. And this is why this is actually a good thing, because when you take on an antibiotic, you want it to affect bacterial cells but not affect your cells, right? So you want to kill off the bacteria but not the human cells. The rest of our structures are really going to be drawn on the outside of the cell. There's not a whole lot, I mean, there's lots of things that go on inside the prokaryotic cell, but that would be more appropriate for a biochemistry class. Those are the 2 major organelles that are inside. On the outside of the cell, we do have some other structures. Right here, this long tail, this whip-like tail, this is referred to as a flagella. Eukaryotic cells have a flagella also. A bacterial flagella and a eukaryotic flagella are actually look the same, but have a different origin. The proteins that make it and the structures that make it up are actually different from each other, even though they look the same. It would be similar to comparing bat wings and bird wings. They look similar to each other because they have a similar function, from the outside, but when you really start to look closely at them, they're really quite different. So we've got flagella right there. Up here, you'll have these long protein spikes that kind of come out from the cell, and those are called fimbriae. Okay. The purpose of fimbriae are actually for attachment. The best analogy I can kind of give is in the old days when they used a lot of ships for transportation and things, they would have, or if you've ever watched, I don't know, like Pirates of the Caribbean or something, when one boat would come up to another boat or they'd come alongside a dock, the boat would have these long hooks with or long poles with a metal hook at the end, and you use that to hook the dock or hook the other ship and pull it into you. And that's very similar to the purpose or function of these fimbriae. They're extensions out from the plasma membrane, and they can grab onto some other surface and help in the attachment of that bacteria to that surface. So if you're in a bacteria, maybe you're living in a fast-moving stream, then you would be able to use those fimbriae to help stick onto the rocks or some kind of substrate inside that stream and hold on to there. If you're in a human body, you could do the same thing. You could use it to hook onto some of the adjacent cells and hold you in place. So that's the purpose of the fimbriae. Down here on the bottom, we have what I'm going to label as a pili or a pili. And so this structure, and many times in micrographs and microscopic pictures of bacteria, you'll see the fimbriae and the pili are often mixed up. In fact, even if you go into the literature and you start looking online, you'll see pili often described as fimbriae or vice versa. The function is a little bit different. The pili a lot of times are a little bit longer than the fimbriae are. And these guys, the pili, have a function in reproduction. And we'll talk more about this in a couple minutes, but their purpose is actually to allow transfer of DNA from one cell into another cell. And we'll come back to that in a second, but that's the purpose of the pili. Now, a couple things that we're missing is, we'll draw a structure right here. And this structure that I'm illustrating in purple, we are going to label as the cell wall that we've already talked about. And what I'd like to do is come over here, and I want to, as if we could magnify, it turns out that in prokaryotes, or bacteria, there are two types of cell wall. So you can take all bacteria and divide them into groups of related species based upon the type of cell wall that they have. And so the two types of cell walls are called gram-positive and gram-negative. And so this is based on a simple assay or a simple test that you can use where you add a gram stain, a fluid called a gram stain, to the preparation of cells. And depending on whether it takes up that gram stain, positive, or doesn't take up that gram stain, negative, is how you classify the bacteria. So if we look a little bit at the structure of this, in a gram-positive cell, you would have, we'll call this layer down here at the bottom, the plasma membrane in black, just as is over there on the drawing that we previously did. And the cell wall of gram-positive bacteria has this thick layer of a material that we already mentioned called peptidoglycan. So here in red we have peptidoglycan. And peptidoglycan is really these long chains of polysaccharides. And these long chains then are interlinked with these short peptide linkers. So that's these little blue dots I'm drawing. And it's a repetitive 3 to 6 amino acids. So it's not really a protein. It's just a short peptide. And they link, these peptides are crossed all over in here. And they link all these polysaccharides together to form the peptidoglycan layer. And that's why it's called peptidoglycan. It's a very identifiable pattern that's repeated over and over to make up that cell wall, the peptidoglycan. And so this would be a gram-positive. It just has a thick peptidoglycan layer. On the other side, you'd have the gram-negative. It also has a plasma membrane. It also has a peptidoglycan layer. But that peptidoglycan layer is much thinner. So over here we have a thick peptidoglycan, and then in the gram-negative we have a thin peptidoglycan. Then we have a new layer right here, and this is called the outer membrane. So gram-negative bacteria actually have a second outer membrane. So this would be a second plasma, not a plasma membrane, but a lipid bilayer. And then in addition, we're going to draw one more piece. They'll have these structures called lipopolysaccharides. Okay? So lipopolysaccharide means that these polysaccharides, they're kind of shorter polysaccharides, but they're attached to a lipid, a special lipid, that's embedded in that outer membrane. And then they sort of stick out like hair follicles from the surface of this plasma membrane. So this is the lipid part, and then we have the polysaccharide tail that's on top of it. So you have lipopolysaccharides, and these are often abbreviated as LPS. Now, some of the reasons why this is significant is that these lipopolysaccharides often act as toxins inside the body. And a lot of gram-negative, not all gram-negative, but many gram-negative are actually pathogenic bacteria. They're pathogens or cause disease or sickness. And the lipopolysaccharides that they contain on the surface are in some cases responsible for that toxicity or for causing people to be sick in them. Okay? So this would be a gram-negative, this would be a gram-positive. One of the things that helps me remember these structures and how I draw them is to look at the gram stain itself. And so the gram stain is actually purple dye molecules. So it's an iodine-based dye. We don't know exactly why or how. We still, after looking for it at it a long time, we don't know exactly why it works. We just know that it works and that you can characterize the cells by adding this purple dye. What I'm about to give you is not so much scientific fact as it is a good explanation that you can remember to help you remember how each type is drawn or illustrated. Okay? So what happens is the purple dye is designed to attach to polysaccharides. And so over here on the thick one, remember gram-positive means that the purple dye did attach to it, right? That's what the positive meant. Well, why would it attach to this one and not to that one? Well, two reasons. One, you've got this really big, thick layer. So if you add the dye, that dye is going to penetrate in in here, and it's going to bind to these. And so you add the dye in the assay, and you let it sit for a while. And you can see how it could penetrate and start binding to these polysaccharides. Then, after a while, you're going to come back and you're going to take some water or fluid, and you're going to wash some alcohol maybe, and you're going to wash this off so that you get rid of the excess dye. And as you do that, it's hard for some of these to get back out, and so they stay in that thick fibrous mesh right there, and it stains the outside surface of the cell purple, gram positive. Over here on the other side, whether this is accurate or not, I don't know, it matters so much, but there's two barriers. One, so you got the purple dye coming in, two problems. In order to get inside and start staining this thin peptidoglycan layer, it has to pass through this outer membrane, which is harder to get through, right? It's not fully exposed. Furthermore, this is a very thin peptidoglycan, so even if it can get in here and bind, when you go to wash it off, it's on the surface, and so it's pretty easy to wash off as well. And so for whatever reason, we find that the purple dye, the iodine-based dye, does not bind to these gram-negative cells. And so hopefully you can use that little explanation to identify and say, oh yeah, this is gram-negative. Negative means there's no purple dye. Why is there no purple dye? Oh yeah, because there's this outer membrane protecting it from the purple dye, and then it's a thin layer, so it doesn't bind very well in that thin layer. If you can remember that little dialogue right there, then you'll have no problem if someone says, well, what's gram-positive look like and what's gram-negative look like, of distinguishing those two types of cell walls. Okay? And so on our drawing, as we come over here, cell wall, it could be a gram-positive cell wall, or it could be a gram-negative cell wall, depending on the type of bacteria. If this were an archaea, we wouldn't even really use the gram stain. It might or it might not stain purple, or the other color is pink in the gram stain. So it might not or might stain depending on its characteristics, but it wouldn't have peptidoglycan in it. So when you looked at the cell wall, the archaea do not have peptidoglycan in it. So that's a little bit about the bacterial cell structures. We learned a little bit about the bacterial genome and about bacterial cell walls. Okay, for our last topic, now let's talk a little bit about prokaryote genetics, and particularly the distribution of prokaryote genetics. And here's the problem that prokaryotes face to some degree, theoretical problem. And the problem is one of diversity. Because single-celled prokaryotes split using a process known as bacterial fission. And in bacterial fission, you just make a copy of that large single circular chromosome, and then you separate them to different sides of the cell, you split it down the middle, and so what you end up with is two identical clones of each other. And so the next generation has exactly the same DNA combination, or very similar to it, as the previous generation. And so there's no diversity as you go through. Now, why is that important? Diversity creates different genetic combinations that allow different organisms to deal with the environment in better or worse ways. And so we often think of diversity, so diversity actually becomes a key to survival. In a given environment, some genetic combinations might be deleterious or they might be disadvantageous, but some are going to be advantageous. And therefore, it always allows life to survive in different environments or situations. And so prokaryotes have this kind of challenge of how do you gain diversity? Now, there's sort of two responses to this. First of all, when you talk about changes in the sequence of DNA, that's referred to as mutation. So mutation is defined as more or less permanent changes in the DNA sequence. And so bacteria are more prone to mutations than eukaryotic organisms are. So they're able to gain mutations, and then so as you copy that DNA, you can actually introduce mutations in the second copy so that the offspring that result from that won't be exact clones of each other, but one will have some mutations. And then, so you can pass on these mutations in what we call a vertical fashion. Humans, eukaryotes over here with their chromosomes, they also will undergo mutation. However, mutation often, well, there can be good mutations, so there can be mutations that provide additional function or added advantages to the environment. They're the minority. So most mutations, if we were to catalog them, would be deleterious or they would be bad for that organism. And so bacteria, the way they get around with this, they just have very rapid reproduction. And so the bacterial strategy is they're just going to reproduce, well, essentially like the saying goes, like rabbits. So they'll have large numbers of offspring. They'll divide themselves very quickly. So bacterial population can double. In optimal conditions, the fastest is about every 20 minutes. Can be taking 40 minutes to an hour for other populations of bacteria. And so they divide very rapidly. And then they have very low concern or low survivability of the offspring. So you might have high mutations and maybe only 10% of the population survives, right? So you have this generation, you double the bacteria, and because of harsh conditions or because they had mutations that were incorrect or deleterious, maybe 90% of that population dies. But 10% of it survives with mutations and is more fit, and the bacterial population as a whole doesn't care because in another 60 minutes, 2 hours, it can gain back the 90% it lost in any single generation. Does that make sense? So it has a high mutation rate and a high reproduction rate and a high mortality rate is what we call it. So a lot of them could die off because of a bad mutation and there's no big loss. Over here, most eukaryotes follow a different strategy. So let's say humans. We don't produce hundreds of offspring and tolerate losing 90% of them, okay? Rather, our strategy is to raise a few offspring but invest a lot of time and energy into having a very low mortality and a lot of training in that offspring. And so we approach diversity from a different point of view. And what we do to achieve diversity is we use basically sex or sexual reproduction, in which we will have one organism, a male, and another organism, a female, and they'll take part of their genetic material encapsulated in the sperm and in the egg, and then they recombine them, and you create a totally new genetic combination in that offspring. It has half of the DNA from the male parent and half of the DNA from the female parent, and you get this totally new combination. And we've mastered through sexual reproduction that approach to not only are we mixing the male and female, but actually within in meiotic cell division, you start to scramble the genes and the chromosomes that actually end up coming together. And you guys will learn about that in another class. But we use sexual reproduction to generate our diversity and pass it on to the next generation. Prokaryotes will use high mutation rates, but they also use another form of reproduction called horizontal genetic transfer. Okay? Now, let me explain. We call this horizontal genetic transfer, and then the opposite of horizontal genetic transfer would be vertical genetic transfer. So let me explain that roughly. If you have some kind of parent right here, and it passes on its genetic blueprint to the offspring, and then you'd pass it on to that offspring. This then would be vertical transfer. We're taking the genetic blueprint and passing it down through generations, and that would be vertical. Horizontal transfer is where you have, we wouldn't really call it a parent, but you have some kind of donor, and it passes it over to another organism that we call the recipient. And so now the recipient receives some new form of DNA, but notice this would be in a horizontal fashion. Okay? In humans, we tend to really think of, and we only really think of vertical transfer, right? So my son or my daughter is going to receive at least half of my DNA in a vertical fashion. In humans, we don't really see horizontal transfer. This would be like me taking a piece of my DNA and giving it to you, and that transforming you, like if you had dark hair and I give you my DNA, and then you'd have blonde hair like me. That would be horizontal, would be two adults, if you will, of the same species or different species even, transferring DNA and transferring the characteristics that DNA encoded. And so the best sort of human example would be when we start to, if you've read up on modern experiments with genetic engineering, right, and fixing genetic diseases or things, that would be the concept of horizontal transfer. And this is a field that's kind of in its infancy, at least with human genetic engineering. We do it quite frequently in animals and plants, but still very nascent for human genetic engineering. However, long before we could do it in plants or animals or humans, bacteria had mastered this idea of genetic engineering or horizontal gene transfer. And there's really three mechanisms that the prokaryotes use for this horizontal gene transfer. So let's over here talk about horizontal genetic transfer. And the three methods are transformation, transduction, and conjugation. So let's go through each one of these one at a time. Some keywords that go with transformation are going to be environmental DNA and a term that we call competent. We'll just call it competence and competence. Okay? And competence is kind of a weird term. It sounds like it's a good thing, but really it's a type of sort of bacterial sickness, if you will. Here's how transformation works. Scientists were able to discover that in a lab you'll have some kind of bacteria, and you could introduce certain chemicals to it, like calcium and other things. And if you grew it in certain chemicals, like a solution that was rich in calcium or some other chemicals, some other salts, different salts, it would affect the growth and development of that bacteria. And it would essentially make it, not really malnutrition, but just kind of sickly. And that when this bacteria became sickly, it caused certain, and again, we're still not even sure how transformation works. We just know that it does. If you add chemicals, grow the bacteria under certain conditions or chemicals, you create components, or what's called a component cell. And that component or sick cell then, if you introduce it to a solution containing what we call plasmids, or DNA just lying naked in the environment, in the solution, that the cell would uptake that DNA. And that's called transformation. Usually it requires a couple things. You have to use component cells, and you have to shock it, heat shock, either with heat, or you can actually electrocute it also. And so you kind of abuse the bacteria in that you make it sick, you make it uncomfortable, and then you stick it in a really foreign environment rapidly, and then it just causes some kind of natural process to occur in the cell. And there's been different theories about, like, the heat shock or electroshock would cause the phospholipids in the membrane to maybe jump apart and allow DNA to pass through. But we really don't know what's happening with it. We just know you make it competent, you shock it, it'll take up DNA from the environment, whatever DNA is out there. So if this were a wild bacteria cell, not in the lab, you have DNA in the soil left over from the death of previous bacteria or organisms, and it would start to take up those pieces of DNA. This might occur, the competence in that wouldn't be from scientists adding chemicals to it, but it would come from environmental extremes, starvation, or something like that would turn on this mechanism and allow the bacteria to search that environment for pieces of DNA that might allow it to exploit and survive in that environment. It might be some genes that allow it to exploit new nutrients in that environment. Maybe it's used to eating and digesting glucose. There's no glucose, there's another sugar, maybe mannose, but it can't digest it. If it pulls in DNA, it might get a couple genes that would allow it to digest the mannose in that environment. Something like that. That's the idea of transformation. We use it in the laboratory all the time, so it's a natural process that we discovered and said, hey, let's use this to do genetic engineering. We'll take the bacteria and we make up the DNA that we put in its environment, tell it to take it up, and now you have DNA inside of that cell in a genetically engineered cell. Okay, let me redefine a term, come back to it, plasmid, because this comes up a lot. What is a plasmid? A plasmid is a small circular piece of DNA. It's different. It's similar to a prokaryote chromosome in that it's circular, but very different in size. So these ones would be anywhere from 1,000 to maybe 10,000 base pairs. Whereas remember over here, our prokaryotic DNA was 1 million to 10 million base pairs. So these are quite a bit shorter plasmids, which is allow them, you'd never be able to get a bacterial chromosome in through transformation. It would be far too big. But these little plasmids can come in. And so plasmids are usually involved, so they carry genes. And those genes are usually involved with improved competition. But they're not required. They're non-essential. So the genes that are contained on a plasmid are usually not ones that are required for that bacteria to leave. If you have a gene that's required, it better be on your genome, right? You better be carrying that around with you on your chromosome. These are ones that just add a better competitive advantage. For example, there might be genes associated, we talked about antibiotics earlier that kill bacteria. Well, wouldn't it be awesome for that bacteria if you had a gene that broke down antibiotics, that allowed it to survive in an antibiotic environment. That would be, again, it would improve its competition, but it wouldn't be part of the sort of housekeeping genes that it would need to survive in most environments. Okay? The example I just gave about the mannose sugar. Again, since it could usually live off of glucose, that might be found on a plasmid as well, genes that might allow you to make use of a different food source or nutrition source in the environment. So that's what a plasmid is, and the reason why we talk about it is because it's often these shorter pieces of DNA that are transferred heterologous or horizontally. Okay? So in our next method, transduction, some words that you should associate right here are phage or virus, a bacterial virus, which is known as a phage. And so transduction involves virus transfer or viral transfer. This one is a little bit more complicated because we actually have two cells. So we're going to have right here, this is bacteria number 1. And bacteria number 1 gets invaded by this phage. And the phage inside it, I'm going to put a little squiggly line right there. That's the phage DNA. And then the bacteria inside has its DNA. And what happens is the phage comes in here, it sticks its DNA. You'll learn a little bit about the virus cycle a little bit in our next lesson. But this bacterial DNA ends up getting chopped up into little pieces. The phage DNA comes in, and it starts to make a whole bunch of new phage particles. Because that's what it does with infection, is it creates new copies of itself. And the idea is that in each one of these new copies of the phage, it would package phage DNA. So you had a new infectious particle, and then that would break out and infect more bacterial cells. But sometimes there's a mix-up. And instead of packaging the viral DNA, it will package one of those pieces of the bacterial DNA. These guys will then break out of the cell, so the cell breaks open, and you have these infectious phage particles. So, the all-purple phage particles, they just go on and infect some other cell. But this unique one that had the bacterial population, let's follow it over to cell 2 that we're going to draw in red. So this now is bacteria 2. It is lucky enough that it does not get infected with an all-purple cell. It gets infected with this purple phage that had the black DNA inside. And so this new bacteria 2, it also has its chromosome in red. Now the phage introduces the black DNA inside here, but this isn't viral DNA. So it doesn't make any new viruses. This cell is not going to die. But it did receive an extra DNA bonus from this cell. This cell had to die in the process, but we now have this new piece of DNA, and that's the horizontal transfer. So it just picked up new genes that it received from a viral delivery system. Okay? This does happen in nature. That's where we first discovered it. And so we'll find remnants of bacterial DNA in a bacterial cell from other species. Sometimes it'll be within the same species, but you can also get transfer across species with certain phages. Just like we use this one, a natural process, but we use it in the laboratory to do genetic engineering, we also use the process of transduction in the laboratory to do genetic engineering. What we do is we simply harvest the phage particles before they've been packaged. And in genetic engineering, so you harvest all the phage proteins, particles, and then you can actually buy these in kits, and you take whatever DNA you want to package, and you dump kind of all the viral proteins unassembled into a test tube, and you dump the DNA that you want it to be assembled with around into a test tube, and it will assemble, it will package, it's called, it will package that DNA that you created into that phage. And the nice thing about it is that the phage then is your delivery system. So you take that test tube of now packaged phages and you pour it into a solution of the bacteria that you want to transduce. And it acts as a very efficient delivery system. It will land on the surface and inject that engineered DNA right into your host organism. So we use viruses because they're efficient delivery systems. This one, you can only get small pieces of DNA, about 10,000 base pairs. Right here, using a phage delivery system, you can actually inject up to 50,000 to 70,000 base pairs of DNA, so it gives you a much larger delivery. We also practice this, although we're talking about bacteria, this is one of the methods that we use to deliver DNA in human genetic engineering also. We use a viral vector, it's called, or a viral delivery system. Okay. So, our last method of horizontal transfer is conjugation. And in conjugation, again, we have two cells. So this time we're going to have some kind of donor cell. And we're going to have a recipient cell. And some words that are associated with this, this would be the closest that bacteria come to what we would call sexual reproduction or bacterial sex. It's also where we talked about that word earlier, the pili. This is where the pili are going to come into function. And then there is something called the F factor. And so the F factor is a group of genes that encode the ability to carry out conjugation. And so you have your donor cell, and it is actually the one that has to have the F factor. So it has a F factor in it, this set of genes, and it will then create this structure that anchors onto another cell, and that's the pili that we drew earlier on our board as we were describing them. And then you'll have a portion of DNA, usually a plasmid. So here's our plasmid. And the plasmid then travels through that pili into the recipient cell. And that's conjugation. Conjugation, unlike true sexual reproduction, it doesn't go both ways. So it's only from the F factor containing donor that passes the DNA. There's no DNA that gets received by the donor, unless this recipient also has an F factor, a set of F factor genes, and then they could exchange both ways across two different pilis. But the usual method is that it's only one way. And so that is conjugation. You have to have two cells, a donor, a recipient, the formation of pili, and then one of the cells has to have an F factor. One other feature, although we've been talking about the transfer of plasmid DNA, and that's usually what it is because the plasmids are small packaged pieces of DNA that travel easily. You can, in conjugation, you can take off small pieces of the chromosome. So you have plasmids or small pieces of chromosome. And again, up here in the phage, in all cases, usually we're talking about plasmids, but particularly in transduction, we may be broken up the chromosome into smaller pieces. And so it might be here that you're transferring not a plasmid DNA, but actually a small piece of the chromosome. Okay? That are 3 different mechanisms of horizontal genetic transfer. In this case, all we're doing is transferring, we're spreading out genetic combinations that already exist. And so we're spreading them out in the population. I'll come back here and reiterate a couple points. The source of all diversity, the source of that original diversity to be spread out, occurred through mutation. Without mutation, you don't have diversity. So mutation is the source of all bacterial diversity. An interesting thing about the horizontal transfer, and this goes along with our idea of genetic engineering, is that the horizontal transfer, particularly in prokaryotes, can cross species boundaries. So you could have one species of cell that will conjugate with a whole different species. That doesn't happen in animals or plants, really, right? You don't see like cats breeding with dogs and mixing genes. But in prokaryotes, you do. You can have you could have whales and chihuahuas, essentially, as far as species differences, be able to recombine. So conjugation, transduction, transformation are not species specific. Okay? They can be used across species boundaries. And that is what we want you to know about prokaryotes. We've learned about the different cell types. We've learned about the different types of prokaryotic cells, archaea and bacteria, and how they're different. We've learned about some of the different structures on a bacterial cell. We've learned about the difference between gram-positive and gram-negative cell walls. And then we've learned a little bit about the genomes, and then mutation and forms of horizontal gene transfer in prokaryotes. We'll see you next time.